Hello and welcome to CTF Connect, a virtual panel discussion. We're so happy that you took time out of your day to join us for this important discussion. I am Hayley Jameson, CTF's founder and president. For those of you who may not be familiar with Clear Thoughts Foundation, often referred to as CTF, we are a local nonprofit started right here in Pittsburgh in 2010 with a mission to fund breakthrough drugs and novel treatments to eliminate dementia, a disease that unfortunately we are all too familiar with. But we are very excited to have you with us today, as I mentioned, and this is a part of our first ever Dementia Awareness Month. So again, excited to be able to offer this educational discussion with our brilliant CTF Consortium doctors. And again, so excited and appreciative of your time and excited for you to actually see what your donations are funding, both our past donors, current donors, and future donors. Again, thank you for joining. And as you can see, I'm joined by our lovely operations manager, who I often refer to as the second half of the dream team. So at this point, I'm excited to introduce Kate Finella, our operations manager.
Thank you, Hayley, for the kind introduction. Hi, everybody. As Hayley said, for those of you that don't know me, my name is Kate Finella, and I have the pleasure of serving as operations manager for Clear Thoughts Foundation. We're very, very excited to have all of you joining on with us today for this educational and informative virtual research panel. In a moment, we're going to go ahead and get started, and you'll be hearing from our three brilliant CTF Consortium researchers. But before I do that, I want to take a moment and introduce to you today's panel facilitator, our very own Chief Scientific Advisor, Dr. Karl Herrup. Dr. Harop is a professor at the University of Pittsburgh School of Medicine. He's also a co-investigator at the Alzheimer's Disease Research Center, also known as the ADRC. And as previously mentioned, he is our very own Chief Scientific Advisor. So we're very, very excited to have him on with us today. Karl, happy to have you here as our facilitator, and thanks for being with us.
Dr. Harop, I believe your mic is muted.
Sorry. Hello, everyone. I'll get this technology straight one day. For those of you who don't know me, my name is Karl Herrup. I am a professor of neurobiology here at the University of Pittsburgh Medical School. Just a bit of personal note, I'm actually a basic scientist, and I've been studying cell and molecular mechanisms underlying dementia for more years than I like to think about. But from that perspective, I can say that we've made a lot of progress about how a healthy brain turns down the path to dementia. But despite all that progress, we have a long way to go. I personally have been working on the problem since 1992, and while my specialty is Alzheimer's disease, my interests are much broader than that, and it's one of the reasons I'm so excited to be a part of the Clear Thoughts Foundation because of the broad view that they take of the problem of dementia. As their Chief Scientific Officer, I've been able to watch and even help guide the establishment of a really innovative approach to looking at dementia as a problem, and that is what we call the Consortium Model. And this all was really the brainchild of both Kate and Hayley, and it began in January of 2020. That really a new type, or at least an innovative type of funding began. So most scientific funding is done through one grant to one individual. What CTF came up with was what they call a consortium model, and that's what you're going to hear about today from our three researchers. This consortium model is really a challenge and an opportunity. The opportunity is that there's funding available to do cutting edge research, but the challenge is that the issues can't be done within the context of a single lab. The issues are to be collaboratively bounced back and forth between the three collaborators you will hear about. And this allows a synergy and ideas to percolate and be examined from alternative perspectives to grow in ways that are maybe just not standard. And so you will hear in a moment about the fruits of that interaction. But as someone who has watched the program develop from its infancy, I can say that the potential is large and still growing. So with that, I want to begin my introduction for our speakers today. But I do want to take a side detour for just a moment and talk about the research environment here over the past year. And it will come as a shock to no one that the past year has been a challenge, not just for all of us in the community, but I think in particular for researchers in a research environment. Because of COVID, our entire research effort was shut down very quickly after the pandemic started, and really only began to slowly trickle open during the late fall. Myself, I was first able to come back to my own lab not until February or March of this year. And while we're opening again, and clinical research, however, is still severely restricted. So I tell you all that because I want you to appreciate that the work you're about to hear about and the progress you're going to hear about was done in a context of which I would call challenging, to say the least. And so the progress and the work that's been able to be accomplished has been done under the most difficult circumstances. So I think as you listen, appreciate that this is really an extraordinary effort that you're going to hear about. So with that, what I'd like to do is turn the rest of the scientific program over to our three researchers, members of the consortium, and our first speaker is Dr. Oscar Lopez. He's a giant in the field of Alzheimer's disease research, and is director of one of the most successful Alzheimer's disease research centers in the world. It's an NIH-funded center and is really one of the jewels in the crown of Pitt research. Dr. Lopez is a practicing clinician, and what you're going to hear today is some of the work he's been doing just in general in clinical research, but then also in a special technique known as plasmapheresis. And so without any further ado, what I'd like to do is turn the virtual stage over to Dr. Lopez and have him tell us about the work that he's been doing.
Thank you for the introduction, Karl. I'm going to start with a sort of a description of the epidemiology of dementia and Alzheimer's disease and why it's so important what we are doing. We know that about 10% of the people after age 65 may have Alzheimer's disease, and half of the population after age 85 may have Alzheimer's disease. And we estimate that in the country we have about 4 to 5 million patients with Alzheimer's disease. And if we don't do anything, this number is going to increase. And now people are talking about 13 million by the year 2015. So that's why it's so urgent to have treatments for this condition. So what we have now, so the classic treatment for Alzheimer's disease is based on a medication that we call cholinesterase inhibitor. And you can see there Tacrin, donepezil, which is the Aricept, rivastigmine, galantamine, and more recently memantine, But after the year 2003, the FDA has not approved any medication for Alzheimer's disease. So we haven't seen anything. And over the past 4 decades, we have there have been more than 200 therapies or interventions that have been proposed or tested for Alzheimer's disease. And when I talk about therapies, I'm talking not only of medications, but also by cognitive stimulation, physical stimulation. So people have been trying practically everything that we know to see if those interventions can help. In general, the therapies are classified as symptomatic. You give a medication, symptoms improve. Disease-modifying therapies is when you give a medication, and the medication has an effect on the biology of the disease. So, for example, in Alzheimer's disease, we know that the most important pathological damage is a deposition of amyloid in the brain. So that's why you have heard about the vaccine against Alzheimer's disease. Those are disease-modifying therapies. And cell therapies are coming up. Typical, for example, cell therapy, it could be a stem cells therapy. So we are very far from those type of interventions. I would say that now what we have are symptomatic therapies and disease-modifying therapies. This is to give you an idea what's going on now. The pipeline for experimental treatments for Alzheimer's disease has never been so rich. We have now 121 agents in clinical trials for the treatment of Alzheimer's disease. And also you can see there, we are also using medications for neuropsychiatric symptoms. And all of these therapies are extremely expensive to develop. It takes a long time to develop a molecule, for example, from phase 1 to phase 2 to phase 3, that it may take 5 to 6 years. And then all these treatments start with an animal model. So you have the basic research that it moves to humans for a small amount and a small number of individuals, then to phase 2, a larger amount of individuals, and then phase 3. So, but one thing that I would like to point out here is that the pipeline for Alzheimer's disease has never been so rich and with a significant heterogeneity of treatments. So what happened there are two, I would say, two important events that happened over the past 20 years. Actually, you can see there, they happened in the year 2020. These are the results of the most recent trials. And as you see, the majority, NS means non-significant, means they were negative. And there were some, they were just, they show a trend, but they couldn't reach a statistical significance. One of the drugs, the aducanumab, I put press release there because we haven't seen the paper, but it showed an effect on cognition and function. And the outcome of these trials now are measured in the amount of the percent of decline. So the percent of less decline with aducanumab range from 22% to 40%. And this drug is now under, the FDA is reviewing the results of this trial, and they said that we will hear about this in early June. The other thing that happened in the year 2020 was the trial of a plasma exchange. And again, you have measures, for example, ADAS-Cog is a measure of cognition, and the ADCS-ADL is a measure of function. So cognition measures memory, and the ADCS-ADL measures, for example, whether people can go and can go to the supermarket or be independent doing shopping. And the other measures are also, I'm going to talk about this in a few minutes, but the plasma exchange, the range of less decline went from 66% to 100%. So this is the technology. It's called the plasma exchange, has been around since 1958, 1960. And basically is the removal of plasma. This technology removes pathological or neurotoxic substances. And it has been used in many neurological disorders and in metabolic disorders. For example, it's one of the treatments for people with primary hypercholesterolemia. And when we remove plasma, we insert albumin, which is a protein. And that's is the most important protein that we have in our body. And I don't want to confuse you with this, all these figures, but if you see on the left, that's the results of the study, of the whole study. And you can see that in pink are the people treated with plasma exchange, and in blue, people in the placebo group. And you can see that there was a 66% less decline in the people treated compared to the placebo. The effect on the mild people, mild disease, was not significant, but it was significant in people with moderate disease. Same thing happened with the activities of daily living. There was a 52% less decline in the group treated with plasma exchange, and you can see with in the mild group we didn't see an effect, but there was an effect in the moderate group. Now, the other measures, these are other measures that of cognition and function. For example, the CDR sum of boxes is a measure of is integrate cognition with activities of daily living, and the ADCS CGIC is measures what's the impression of the physician on the response to the treatment. And you can see that in the CDR, there was a 71% less decline in the people treated, and this remained like this in the mild and the moderate stages of the disease. In the ADCS and CGIC, which is the impression of that physician has on the patients, the less decline was 100%, which means there was no change with baseline. And in the people with mild disease, they improved. And in the people with moderate disease, there was a 64% less decline. Now, we are using a technology that has been around since 1958, and we know that it's working, but we don't know exactly why. Is there an effect on the biology of the disease, on the amyloid, or is there anything else that is driving this significant response? And here is where the Clear Thoughts Foundation came in, and we are doing the plasmapheresis in a mice. And we can do the same technique that we do in humans. We can do it in mice, and the advantage is we can take pictures of the brain of the mice when we're doing the plasma exchange. So we have a window in the brain, and we can take pictures of what happened in real time of the plasma exchange. And so far what we have seen is that the mice treated with plasma exchange had decreased the position of amyloid, and the brain perfusion is faster in these mice. So we are gradually trying to understand the effect of plasma exchange, and it seems to be that is on the biology of the disease, but also improving perfusion of the brain, so the blood flow. So we are entering in a new era of Alzheimer's disease treatment. So most likely we will have a combination of treatments. So we don't know exactly when, but there are multiple drugs that you can see that they're coming, and we will be using a combination of treatments. The cholinesterase inhibitors that we have had for the past 40 years, they are going to be at the center of the treatment. They will remain, and we are going to keep using them. And there are significant efforts to understand the AD pathological process, and understanding this is critical to develop effective treatments. And thank you very much. And I would like to thank the Clear Thoughts Foundation, because it's at the center of this new technology. It's helping us to understand how an effective treatment is working. And thank you, Hayley.
Thanks, Dr. Lopez. That was absolutely spectacular. And as someone who's been in the field for a long time, it is really thrilling to see how the clinical, how big the clinical impact of this new technology is. And so without saving questions and comments until the end, I'd like to move on in our look at the work of the consortium, and introduce our next speaker, who will be Dr. Amantha Thathiah. She is also a member of the neurobiology department here at the medical school, where she is a rising star, and also a member of the Pittsburgh Institute of Neurodegenerative Diseases. Her interest in research and in dementia has focused on a microtubule-associated protein known as tau, which I'm sure you will hear quite a bit about. But I think the work you're going to hear is just as exciting as the work from Dr. Lopez. So, Dr. Thathiah, the floor is yours.
Thank you, Karl. Can you see my screen?
Yes.
So do you see it now in presenter? Yeah. Good. Okay.
Good.
Okay. So first of all, thank you, Karl, for the kind introduction, and thank you, Clear Thoughts Foundation and Hayley, for giving me this opportunity. So I wanted to first begin with a simple question. Well, I don't think it's a simple question, but what is dementia? And so I think we know now that dementia is not one specific disease, but it's an umbrella term that describes a collection of symptoms that are caused by disorders that affect the brain. It affects thinking, behavior, and the ability to perform daily tasks. And brain function is affected enough to interfere with the person's normal social and working life. And Alzheimer's disease is the most common form of dementia, but frontotemporal dementia also represents 5 to 10% of all cases. So from that, I'd like to transition into what are tauopathies. So in Alzheimer's disease, we know that there's an accumulation of amyloid plaques in the brains of patients, but there's also a group of neurodegenerative disorders that are defined by the presence of abnormal aggregation of this protein called tau. And this is characteristic of Alzheimer's disease, as well as frontotemporal dementia and other tauopathies. Okay, so I guess I study basically post-mortem human brain samples, and then I transition what we see in the brain of these post-mortem brain samples into trying to test them in vitro models, in cells and in mouse models of Alzheimer's disease. So in the normal human brain, if you take a slice of the brain, there's this region called the hippocampus. And this hippocampus is very important for learning and memory. And the neurons in a healthy brain, they look nice. They have these nice processes with these little stub-like things on them. However, in the diseased brain, you see this shrinkage of the brain, you see enlargement of ventricles, and a dramatic shrinkage in this hippocampal region, and also a lot of neuronal loss in this region as well. So in my brain, in my lab, what we do is we take slices of the hippocampus, and so this is a slice of the human hippocampus, and in green you see neurons. And they're stained for just a marker that recognizes neurons in the brain. And so this in green here is a healthy neuron, and in blue is the nucleus that contains your genetic information. And to the right of it is a dying neuron, and this dying neuron is overburdened with tau. And I was trying to come up with a nice analogy from everyday life as to how to think of the accumulation and aggregation of tau in the brain. But the only analogy that I could actually come up with was that if you think of your brain as your kitchen, and you think of a neuron as the sink, so basically you put dishes in the sink and think of this as tau, and you clean it. If you have enough soap, you have a good drainage system, you clean it, the dishes get put away. But what happens in Alzheimer's disease is these dishes, for whatever reason, the drain get clogged, or you don't have enough soap, the dishes start to accumulate, and so tau starts to accumulate in your sink, basically, and it starts to overflow, and this starts to cause neurons to die, and which results in cognitive impairment. Okay, I tried. So if we look at this in more detail, you can again see this healthy neuron, and in this neuron you have a certain amount of tau there that's important for some normal physiological functions. However, in disease, it starts to accumulate these P circles, which are phosphorylation. So it starts to become phosphorylation, hyperphosphorylated or excessively phosphorylated, and this can lead to tau molecules to start to stick together. So it starts to form dimers, 2 molecules sticking together, 3 molecules, or multiple molecules of tau that start to stick together, and they start to aggregate and form these NFTs or neuronal tangles that are then present and what you see in patients with dementia and Alzheimer's disease. And this leads to neurons to eventually die. Okay, so we don't know what causes tau to aggregate, accumulate, or cause cells to die in the brain. And so, but in cases of the human brain, what we know is we see healthy neurons, we see dying neurons, but we don't know how they go from a healthy neuron to a dying neuron. And so we need to develop in vitro systems to be able to study this. Okay, so what my lab thanks to the grant from CTF Foundation, there's this protein called VIVID, and it's found in fungi. And when it's exposed to blue light, it starts to aggregate. Okay. So what we did is we fused this VIVID protein that's prone to aggregate to tau, and we formed this molecule called opto-tau. And so basically, it's just a light-induced tau model. And so when we shine light, blue light, on tau that's attached to this protein called VIVID, we can then promote tau aggregation. And then what we've been hoping is that we could then form some tau aggregate structures that perhaps resemble the tau aggregates and the neuronal tangles that are found in AD or dementia patients. And so how do we study this in vitro? So we take cells, just a normal cell line that we use, everyday cell line that we can use in labs that we can propagate indefinitely in culture. We introduce our tau VIVID molecule to these cells. We shine blue light on these cells to induce the aggregation of tau once it's expressed in these cells, and then we extract the protein from these cells. So basically, we form a fruit smoothie of all the proteins that are found in these cells. We then separate these proteins on a porous gel matrix, and these proteins diffuse along this gel matrix according to their weight. Heavier molecules you would find at the top of this gel. Smaller molecules you'll find migrating more faster towards the bottom of the gel. And what we can then do is probe these membranes with an antibody that specifically recognizes tau. So high molecular weight tau or tau that's aggregated is going to run closer to the top of the gel, and we can track that. And we can see, if you notice here, this is tau in the dark that hasn't been induced to aggregate, so you don't see much of this aggregated tau. However, when we expose this opto-tau molecule to blue light for 1 hour, or 4 hours, or 24 hours, you see an increase in this dark band, so this aggregated species of tau. And we can also then look at this monomeric, single monomeric tau molecule that hasn't aggregated in the sample as well. And so what this data shows us is that we've developed a system to nicely, in a temporal way, a time manner, induce the increasing aggregation of tau in vitro. And so along with this, what we can also do is we can take these cells, we can label them with an antibody that recognizes tau, and then add a fluorescent tag to that, and then using microscopic imaging, which some of you have seen when you came to visit my lab, we can then now look at these aggregated tau species in cell culture model. And so using this model, we can now ask very specific questions about what causes tau aggregation, how does it aggregate, how can we perhaps stop tau from aggregating. So just a brief summary of the research that we've been doing with CTF. So we know that tau aggregates in cases of dementia, such as frontotemporal dementia and Alzheimer's disease, which leads to neuronal death and cognitive decline in patients. So why tau aggregates and what causes tau to aggregate are unknown. However, this is fundamental knowledge that is necessary to understand disease mechanisms. So my lab, with the help of Clear Thoughts, has established a novel approach to study the aggregation of tau. And this is a very powerful way to induce tau aggregation in potentially a spatially and temporal manner. And so moving forward, we're currently investigating factors that may induce, accelerate, or slow tau aggregation. We're also investigating inhibitors of tau aggregation, which could be a potential platform for therapeutic testing of drugs that could potentially inhibit tau aggregation. And also, in collaboration with Dr. Friedlander's lab, an area of research that I've studied for many years has been G protein coupled receptors. And so we have teamed up to study GPR3, my favorite G protein coupled receptor, and I think his favorite GPCR, the melatonin receptor, to understand mitochondrial function in an Alzheimer's disease model. Okay, and so with that, I'd like to thank my lab, and especially Clear Thoughts Foundation. And I guess thank you for attention, and I'll welcome any questions at the end.
Thanks, Dr. Thathiah. That was great. And as you can see, the research is different, and yet you're beginning to see the linkages among the various groups. Again, we'll have time for question at the end, but right now what I'd like to do is move on to our third and final scientific presenter of the day, Dr. Robert Friedlander. He too is a major figure in the field of dementia research and a contributor to Clear Thoughts Foundation in many, many ways. He's also the chairman of the University of Pittsburgh Department of Neurological Surgery, and he's the co-director of the Neurological Institute. And you heard a hint that some of the research we're going to hear about is based on the sleep protein, melatonin. But I won't take up any more time and turn the virtual podium over to Dr. Friedlander for his talk.
Well, thank you very much, Karl. Can you hear me and see my slides?
Yes.
Okay, thank you. First of all, really wanted to thank the foundation, particularly Hayley, her family, and her board, Kate, all the great work that everybody's doing for getting us together and assisting with very, very important funding, which is obviously critical to be able to do all this work. What I'd like to do is provide an overview of what my lab does. Obviously, with a goal of really targeting it towards dementia, and what does this all mean from that mechanism. I'll talk a lot about the mitochondria, which is an organelle within the cell. So an overview is really that a common feature of many neurological diseases is neuronal death. And in my lab over the years, I've been focusing mostly on the work on Huntington's disease, ALS, and stroke. And one of the beauties of this consortia is that is to be able to import people with other types of knowledge into the field of dementia. So that's been really terrific for me and for my lab. So again, neurological diseases, the key feature is neuronal cell death as it occurs in all forms of dementia. As you know, for the most part, the brain and spinal cord have very, very limited ability to repair and regenerate, particularly as we age. So to inhibit or to block the process of cell death is key to really develop a therapy for these diseases. Now, understanding the process of both normal and pathological cell death, my work really has targeted the mitochondria, and I'll be speaking more about this. So really, our goal is to develop therapies to stop cell death. Now, what is the mitochondria? The mitochondria is an organelle, is a part of the cell, and is critical as a target for neuroprotection. So normally the mitochondria is, you can think of it as a nuclear reactor. When it works well, it's fantastic, but when it doesn't, is a disaster. So mitochondrial function normally, it results in energy production, and a healthy cell has a lot of energy. Now the other part is within the mitochondria, and it's unclear why. There are a lot of different factors that once and if they're released from the mitochondria, they actually promote cell death and promote inflammation of the brain. So when the mitochondria does not work well, there's a bioenergetic collapse. You release all these activators of cell death, free radical generation, and you activate the inflammation within the brain. So just as a cartoon here, very briefly, inside the cell in green, you can see the mitochondria. And if something bad happens to the cell, and that could be, again, a broad variety of different issues. You have changes around the mitochondria, like what's called BCL2 members or calcium changes or free radical. And then what happens is you release all these factors from the mitochondria, which then go on to trigger the cell death pathway. So if you can block this process, you block the process of cell death. Now, what I'll go is I'll take little snippets of some of our work and how it's relevant to what we're trying to do here. Borrowing from our work in Huntington's disease, what we found is that the Huntington protein, what it does is actually it clogs up the mitochondria. Again, going back to the clogged sink analogy, it clogs up the mitochondria, and you get backup of all different problems. And this is one thing that we found, but we only found this in the processes all the way at the end of the neuron. So this is a picture in a tissue culture of a normal neuron. Green's just a marker for the cell. Red is our mitochondria that have energy. Mitochondria can have energy or lack energy or gradation from this process. So as the cells die, the first thing that happens is the mitochondria lose energy, and what you could see is that all the way at the end of the cells, it starts degenerating, the synapses that Amantha described as well. So all the way at the end is the most vulnerable part of the cell, and the question is why? And one of the findings that we identified within the last couple of years is this process of the energy of the mitochondria. So this is a picture of a perfectly normal neuron that's living happily in the tissue culture dish. What's marked as red are mitochondria with high energy, and what's marked as blue are mitochondria with low energy. What you could see in the center, mitochondria have high energy, and the further out that you get from the center, there's less energy in the mitochondria. So this is a perfectly healthy neuron. So this is a vulnerability that our neurons have. So all the way out at the end is where the cells are the most vulnerable. And what I'll show you here is a little video All the mitochondria are colored green, and there's a dye in it that colors them red if they have energy, and they lose the red when they don't have energy. So when they don't have energy, it's green. And what you could see is that the neurons are going from the nucleus, which is on the right side of the soma, all the way out at the end. So the ones that are going out have energy, and the ones that are coming back don't have energy. So again, it's this transfer of energy, and understanding the basic biology of this has helped us to understand this process of why the mitochondria all the way out in the processes are more vulnerable. So what we found is that if you have the mitochondria, when I say I have a stress signal, stress could be aging, could be free radical damage, could be Huntington, could be an ALS. The mitochondria swell, it releases all these factors which activate the cell death process. And a drug that we worked years ago, minocycline, the antibiotic, actually blocks this process and protects the neurons. So what we wanted to do is identify, are there other drugs that do the same thing, that can protect the neurons? And the NIH put out what's called an RFA as a request for applications. Ours was approved, and without going into the details, we did a mitochondrial screen, and what we found a number of drugs that block this process of cell death, but one that really caught my attention was melatonin, for a number of reasons. Again, melatonin is, you've all heard of it. You can buy it in the store. Has many functions. The most commonly known is that really it helps you sleep. It affects the circadian rhythm, but within the brain, and this is some of the work that we've done more recently, it blocks the production of free radicals. Free radicals damage the brain, improves mitochondrial function, which again is something that interested me. It blocks a lot of these cell death processes, and when we use them in mice, in models, it's protective broadly in stroke and Huntington's and ALS and Parkinson's and so on. Now, the one thing that we found is that the highest concentrations of melatonin are in the mitochondria. And again, without going into all the details, actually melatonin in the neurons is made in the mitochondria and only made in the mitochondria, which is something very interesting. Now, as we get older, we lose the amount of melatonin that is made. As you can see, this is just levels of melatonin throughout the day. At night is when you get the spike of melatonin. And when we're young, we have a higher spike. When we get older, there's less and less. So that's why sometimes, as we get older, it's more difficult to fall asleep, or we wake up earlier. But also importantly, we're losing the protective properties of melatonin in our brain as we age. So the new research paths that are being funded and incented by CTF are, one is we created a cell line that does not make any melatonin, and we were actually using this cell line as aging in a dish. We're screening for drugs that block this process of neurodegeneration. We also created a mouse model that does not make melatonin. So again, that's aging in vivo. So this mouse is providing us a tool to evaluate the aging process and how we can slow down the aging process, which again, obviously is a clear correlation with dementia. And as Dr. Thathiah also commented, we're working jointly in a new Alzheimer's mouse model, and we're collaborating, again, looking at these mechanisms of cell death, particularly as they pertain to melatonin. So as I sum up here towards the end, what we found is that there's a two-hit neurodegeneration hypothesis, where one, as we demonstrated initially, the neurons all the way up in the processes, the mitochondria are more vulnerable. And that, again, that's the first site of vulnerability. Then anything else affects you where you have the least amount of energy. So that in Alzheimer's could be expression of any of these mutant proteins or any of these other diseases, and that's where the process occurs. So for future goals, we want to use these two powerful tools to really understand the aging process in collaboration also with the Drug Discovery Institute at the University of Pittsburgh. We are developing a high throughput assay to evaluate for new drugs to halt neurodegeneration. So in conclusion, again, the mitochondria is a gatekeeper of cell fate. Understanding mitochondrial function and dysfunction in the brain is key for development of new therapies for aging, for dementia and neurodegeneration, and obviously neuronal cell death is a common shared feature among all these diseases. I want to thank some of our sources of funding, obviously in particular the Clear Thoughts Foundation. Really appreciate all the work that you do for us, as well as the people that work in my lab. So again, thank you very much for your attention, and we look forward to questions.
So thanks so much, Dr. Friedlander. Let me just put in just a few comments because I want to leave plenty of time for questions from the people who are attending. And I think what you heard today is how exciting and rapidly the field is advancing and how progress is being made. But I hope in some ways that the subtext of what you heard was that there is a lot of work left to be done. And we are making progress, but there is a long way to go. So with that, let me close the scientific session. And at this point, I'd like to turn the virtual podium back to Justin Meyer, who will handle the question and answer period.
Thank you, Dr. Harop, and thank you all, Dr. Thathiah, Dr. Lopez, Dr. Friedlander, and Hayley and Kate, and all the board at the foundation. What a really fun day here. So we have a lot of questions, a lot of them. So we'll try to get to as many as we can in our allotted time here. We're going to start with Dr. Lopez. Dr. Lopez, a question. When you refer to an increase in brain perfusion, what do you mean exactly? Does plasmapheresis improve blood flow to the brain? How is this important?
Well, it's important because improving perfusion means more oxygen to the brain and better nutrients to the neurons.
Very good. Thank you, Dr. Lopez. I'll keep you on the screen here. I think we have 2 more questions that we'll throw at you while we have you here. I have dementia in my family. Is there something I can do to test my potential to suffer from it and for any resisting procedures that might reduce my risk?
Well, you mentioned very well risk. You can have sort of a genetic testing. People are now, they go to one of these commercial places where you can be tested for the apolipoprotein 4, but carrying the allele of APOE4 allele, which is the risk, the genetic risk, is a risk. It doesn't mean that you're going to have it. So we have to be very careful when somebody's telling us you have an increased risk or you are for sure you are going to have dementia. So far what we have are elements that can tell us something about risk or something that is going to happen in the future, but it's not a done deal.
Thank you, Dr. Lopez. Next question is for Dr. Thathiah. Is it possible for scientists outside of Pittsburgh to collaborate with Dr. Thathiah to use her platform technology to test their agents to treat dementia?
Of course, I welcome collaboration, very open to it. That's the only way we can advance science and research, I think.
Good. Thank you, Dr. Thathiah. Next question for Dr. Friedlander. Dr. Friedlander, looking to the future, what type of advancements do you hope to see arise from your collaborative research within the next 2 to 3 years?
Well, what's important with the dementia, Alzheimer's, and a lot of these neurodegenerative diseases is that, as you know, there are many things happening to the cell. The cell gets exposed to so many different problems, and I believe that we're going to need a combination type of approach, similar to what happened in HIV, where at first it was a death sentence, and then there was one drug, and now there's many drugs, and really this cocktail type of approach where there are many drugs targeting different aspects of the pathologic process it's a chronic disease. It's changed completely what's happened, and that's what we need to do in dementia and in neurodegenerative diseases, where hopefully we find different opportunities. I think, Dr. Lopez, what he described today is incredibly exciting and really a game changer for Alzheimer's. And as we develop additional therapies with different drugs, each one of the drugs by itself probably would have a small effect, but once we start to add them together, that's when we're going to see a significant effect. So really, to have both experimental models and a rapid way of testing them in humans, they're new trial designs now that people are developing to really to be able to do combination type of therapies and to test these in a more effective manner.
Thank you, Dr. Friedlander. A couple other questions popped in here while you were speaking. Could you speak to how sharing information and taking a collaborative approach to your research has worked under the CTF Consortium? And
sorry, that was to me?
Yes, I'm sorry. Yes.
Yeah, so I think it's, as we mentioned today between the three of us, there's been significant back and forth, and again, CTF has lubricated, I would say, just putting us together, and it's been wonderful with Dr. Thathiah. We have significant aspects in common from the pathways that we evaluate. They're different, which is nice, but they're complementary, and we're certainly talking about working and collaborating, and even beyond the work that's done with the CTF. But this will certainly have an impact. And with Dr. Lopez, we've put together applications actually to test melatonin in Alzheimer's disease. It hasn't been approved at this stage, but we're working together and we're thinking about how we can collaborate, and the sum of us makes us all better.
Thank you, Dr. Friedlander. And one last question that came in about melatonin. Do you think that melatonin production in the brain is important for protecting other parts of the body from the effects of aging?
I think yes. One of the things that happens not only in the brain but throughout the body, as an example, is a free radical damage. And melatonin is a very, very potent free radical scavenger, which again neutralizes a lot of these free radicals. So as the damage amounts as we get older, melatonin can ameliorate that process.
Thank you, Dr. Friedlander. Next question is for Dr. Thathiah. Dr. Thathiah, what makes your dementia-related research unique to that of others across the country and world?
Wow. Okay. So it's unique in that nobody has developed an in vitro model to study tau aggregation. Other systems have used different forms of tau that have mutations in them, but it's been incredibly difficult to study the aggregation of just normal wild type tau that you find in the brain. And so the research is novel and unique in the fact that we now, for the first time, can at will basically induce the aggregation of tau. And with that, we can now ask very, I think, physiologically relevant questions in the context of the tau molecule that is actually expressed in the brain of AD patients.
Thank you, Dr. Thathiah. Next question is for Dr. Lopez. Dr. Lopez, are there any preventative practices that I can do now to help eliminate the risk of developing dementia?
Well, so we need to take care of the risk factors, and there are many papers published that have shown that the most important risk factor is hypertension, for example, diabetes. So taking care of your blood pressure, I would say, is the most important thing that you can do. And if you have diabetes, controlling your diabetes. So those two things are critical. And because they can trigger, we don't know exactly how, but they can trigger what the release of these proteins or factors that can alter mitochondrial function, for example. They can alter the neuronal function. So we don't know exactly how they connect, but we know that having increased blood pressure can determine abnormal cell function. So it's a very simple thing. Keep your blood pressure under control.
Thank you, Dr. Lopez. Very interesting. Looks like we're going to have room for one more question. I think it's a good one to wrap up with, Dr. Lopez. What can I do to help make a difference in eradicating this disease? And I think we talk about it a lot in philanthropy, and being on this presentation today with a great foundation that does a lot of great work. Dr. Lopez, could you talk about the importance of philanthropy for research in this disease?
Well, there are two things that people can do. One is volunteer for research studies. Being sometimes a normal control in a study, in any study, is extremely important. And philanthropy has a significant role because it helps researchers to develop new ideas. So when you submit a grant to the NIH, basically what you have is something that is already took off. So philanthropy is critical for the development of new ideas, and that's it is just is the, and from there you go to these federal agencies, to these large foundations. You can ask for funding, but I would say the generation of ideas and innovation is with philanthropy.
Thank you so much, Dr. Lopez, and thank you to all the researchers. Thank you to Karl. I'm going to throw it over to Kate and Hayley to wrap it up for us today.
Wonderful. Well, thank you so much, Justin. Unfortunately, we have reached the end of our time together today, but I just want to thank you all one more time for joining on with us today to learn more about our exciting dementia research that's happening right here in Pittsburgh. I also just want to take a moment to thank you, Justin Meyer, for your coordination and hosting of this event. To thank yourself, Dr. Karl Herrup, our Chief Scientific Advisor, for facilitating, as well as our three brilliant researchers, Dr. Friedlander, Dr. Lopez, and Dr. Thathiah, for sharing these wonderful research advancements with all of us today. If any of you on the call are interested in learning more about the CTF Consortium's research, how you can participate in our Dementia Awareness Month for the duration of May, or ways you can support our mission, and much more, please visit our website at clearthoughtsfoundation.org. So at this point in time, I'm going to turn it back over to our founder and president, Hayley, to see if there's any closing thoughts.
Great. Thank you, Kate. And again, I too wanted to thank all of you for joining us today. As I mentioned earlier, we certainly are grateful for your support and taking time out of your days to learn about the important work being done right here in Pittsburgh, thanks to your donations. And I don't know about you guys, but I feel like I should raise my hand because I think I missed too many biology classes in my college days. Some really brilliant work being done. And again, we're so very grateful for all of the work and your time today, to all of the doctors and researchers and scientists that are part of CTF Consortium, as well as Karl, to you. So again, thank you for being part of the call. I would ask for folks that are interested in helping us fund the fight against dementia to stay connected with CTF. We do our best job to get all of our events and activities out there, both on our website as well as social media. Our big annual gala is coming up. It's always the first Saturday in November. So this year it's Saturday, November 6th, and it'll be in person. Woo, 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 woo. Last year, as some of you may know, it had to be virtual. So we're excited to get back to in person, and it's at J. Verno Studio on the South Side. So for those that are local, please join us. And for those that aren't local, hop on a plane and join us. So, so mark your calendars, November 6th. But again, on a serious note, I am so very personally grateful for all of your time and all of us on the board. We have an incredible board that works relentlessly behind the scenes to help with all these events. So thank you to our board. Thank you to our viewers for tuning in. It is a devastating disease. Unfortunately, I've just, my family has been personally hit again with dementia. So, and I know millions and millions others have had that same misfortune happen to their family. So please continue to help us fund the fight against dementia. Thank you again for joining in. Hope to see you in person on November 6th, and have a great day. Thanks.
Bye.
Thank you.
Bye-bye.